many years ago, and some of the little things that we are doing after that work, and where we think that this research, will be heading in the future. So the reason why I think that looking at the structures of polyproteins was really an enigma in the beginning, in all honesty, and we absolutely had no idea what to expect, and whether we are going to see things properly folded or not, So, but it took a bit of effort to also get it done, and so that's why I thought that at the beginning we were actually chasing an enigma, and thankfully we succeeded. So as of 2005, these are global statistics of people living with HIV. There's about 40 million people, almost 41 million living with the disease. But sadly, more than 600,000 people die from HIV every year. And this is despite the drugs that have been discovered over the years and which have been really, really great in helping people live normal lives. But even with all that, HIV-related deaths are reaching 700,000 soon. And that's something that means that we need better drugs. We need new interventions that would help compliance and all the other things that make people not get improve access as well. And so if we are going to be able to develop new drugs against HIV, it means that we need to look beyond the current targets that we've been looking at. And so that is where we started from. So we go back to review the life cycle of the virus and see where we could potentially target in terms of being able to develop new drugs for HIV. HIV infection starts with the virus, a mature virus, we call it, on the surface of which is an envelope glycoprotein, two heterodimer, GP120, GP140, the transmembrane domain and the outer domain. When the GP120 engages immune cells, basically that have CD4 on their surface, the GP120 engages or binds the CD4 receptors. Conformational change occurs in the GP120 that allows the transmembrane domain to also engage co-receptors, typically CCR5 and CXCR4 on these immune cells. That allows the membrane of the new virus to fuse with the new ones, releasing the core of the virus into the cytoplasm. When the core is released into the cytoplasm, it gets transported through microtubules or whatever cargo it is into the nucleus. Now, it is believed that in the course of the transport of the Fullerene cone structure into the nucleus, reverse transcription takes place where the single-stranded RNA genome gets copied into a double-stranded DNA. The core gets docked at the nuclear pore complex and gets deposited into the nucleus, where disassembly of the capsid takes place, and that integrates, picks up the double-stranded DNA, and then inserts it into the host genome. From there, transcription of new RNA takes place, which gets exported into the cytoplasm, and the RNA gets translated into concatenating proteins we typically call polyproteins in the cytoplasm, and then these are transported to the plasma membrane where new virus begins to assemble. Now, I want you to take note of these polyproteins because we will return to these soon. When assembly is complete, the new virus batch off the surface into an immature virus where under microscope, it appears spherical. During that process, a protease which is encoded in the Paul gene gets activated, and that protease goes about cleaving all the polyproteins that have been produced from the viral RNA. And the capsid that is produced thereafter rearranges itself into this fullerene cone. And now the virus is ready to reinfect new cells. If you look at the HIV genome, it's very interesting. There is a single genome for all the proteins that are encoded. At the 5' end, the structural proteins, we call them GAG, and the 3' end are the enzymatic proteins, we call them POL. And so from a single mRNA, the structural proteins and the enzymatic proteins are produced. And these normally happens through a -1 ribosomal frame shift. This frame shift happens at the C terminal, at the 3' end of the P6. So as the ribosome is going, it gets to the end and it encounters this stem loop, which is very stable, coupled with the energetically very weak bases over here. The ribosome slips back and forth, and that allows the -1 frame shift to happen. Incidentally, the ratio, and that produces either a GAG when it truncates over here or the GAG pore when there's a read-through. Now, incidentally, the ratio of GAG to GAG pore is at 20:1. So pretty much you have 5% of GAG pore inside the virion. Interestingly, you can modulate the stability of this stem loop to produce different ratios of GAG to GAG pore. Now, if you increase the GAG pore ratio, it turns out that there is premature activation and therefore infectivity is lost. Now, this we believe is very important for the generation of next anti-HIV drugs, and that's something that we'll be looking at down the line. A cousin to HIV is the foamy virus, and these are really the most ancient viruses, but we call them unconventional because unlike the classical retroviral genome that I've just shown you, they don't produce their structural and enzymatic proteins from the same mRNA, but rather the structural proteins and the enzymatic proteins are produced through alternative splicing. And so the GAG gene is produced from a GAG mRNA, which is spliced from the GAG pole or from the genome. And while the pole is also spliced from the genome, and so the GAG polyprotein and the pole polyproteins are produced. Incidentally, only about 3 kilodaltons is cleaved off the C terminus of the GAG polyprotein for foamy viruses, making the mature GAG virtually almost identical to the immature one. For the pole, only the integrase is cleaved off during maturation, and so the functional reverse transcriptase also has a protease attached to it. And so looking at all the years, over the years, all the work that has been done, we have solved all the structures of the mature proteins, and these have been very instrumental in developing, new drugs. But how do the immature precursors look like? This is a question that we've been very interested in for a very long time. We have absolutely no idea. And so we were interested in finding out how these immature precursors look like. Again, like I mentioned, almost close to 30 drugs for HIV, all of them target the mature enzymes. And so we thought that, well, if there is any possibility that these immature enzymes are ordered in some way, we could use them for developing new therapeutics. And so in the field, there are very critical questions that we needed to answer, some of which we still don't have answers to. And that is, for example, how are retroviral proteins arranged when they are in the immature precursor? Protease dimerization or activation is required for the maturation process. And so the question is, how does that happen? Do we have any architectural differences between the mature protease and the immature one? And how does it affect substrate binding and stuff like that? There is reverse transcriptase, there's integrase in the pore. How do these look like really when they are still all together as a polyprotein precursor? And how do these multimeric states of the RT and integrase influence protease activation? These are very critical questions that we needed answers to. And so um, just to avare your minds too, we're not the only ones who were interested in answering these questions. And so Bob London and his group at the NIH, they have been trying using various isotopic labeling and NMR to try to figure out how this process happens. And so this is one of the models that they published in Viruses back in 2016. They were thinking that, well, the protease, first of all, dimerizes and then integrates may do so. And then once the protease is dimerized, it will cleave off the integrase, and that will allow the reverse transcriptase portion to also dimerize. And then the protease can cleave the C terminus of the P51 RNase to now mature the RT, and then subsequently the protease cleaves itself. So this is how they were thinking about based on some of the experiment that they did. But of course, because we didn't know anything, I mean, these were all ways by which one could visualize what could possibly be happening in the polyprotein precursors. And so we started off by going back to the foamy virus to see whether that is going to be easier to deal with. And so we took the wild type protease reverse transcriptase, which is the mature form of the reverse transcriptase for the foamy virus, and try to work with it. Well, we're a bit successful in that, I mean, you could purify the wild type enzyme, but was very laborious. After 3 steps purification, this is about the best shot that you could get. And you always get this band around 65 kilodaltons coming along with the major band. Of course, for structural work, this becomes a very serious problem or creates complications for us in terms of crystallization or even cryo-EM. But also, we needed to use a lot of protease inhibitors to even get to this level of quality. And so the question that we're asking ourselves, is there a way of getting rid of this? In fact, using biophysical techniques, all the purification methods, you can't get rid of this guy. And so question we were asking, how do we identify where this proteolysis is happening that we could probe and then say make mutation over there or something like that to stop this proteolysis from happening? And so one of the things that I did was to copy the sequence that I was working with at the time into XParsy to see what the instability index prediction was. I was surprised that the instability index was predicted to be 38.83, which is at borderline stability. And so I felt that, well, okay, it means that we could help make the protein a bit more stable. But here we are, we didn't know exactly where the cleavage was happening. And so we wanted to find out where is the proteolytic hotspot in the protein that we could make mutations to stabilize that as an option. And so at the time, I asked my roommate who was in computer science, and discussing with that, Jan, he wrote a script for me that allowed me to computationally mutate all the residues in the protein. And when I mutate, I would replace it with the other ones. So if I take amino acid 1, I will replace it with all the other amino acids and then output an instability index as an option to see whether we will find a place where it's more, others are more preferable to that. Thankfully, we found that at H507 and S584, these appear to be the hotspots from the computational perspective. And so with a trick that we had used, C280, which is a surface exposed cysteine that also causes some aggregation. So I mutated the C280 to a cysteine, a serine, sorry, and then mutated H507 to a D and S584 to a K. And to our surprise, that protein was very well expressed. You could purify tons of it from a liter of prep. In fact, you could get more than 50 milligrams of purified protein, with a very simplified, workout without even you needing any protease inhibitors. And the protein was intact. You didn't have any issue with a breakdown. And so this was a really nice, for us to get intact protein that we could use for our various crystallization efforts to try to get to the structure. Well, once we've made mutations in there, it was important to test whether the protein we are working with is active or not active. And so our collaborator, Steve Hughes, of the NIH, he's always been the go-to person when we wanted to do these assays. And so we sent it to him. Thankfully, both RNase H activity and polymerase processivity was fine. I mean, compared to the wild type, the mutant had it was a bit weak, but again, it was active. And so we knew that, well, we probably haven't completely obliterated, the structure in a way that doesn't support activity. And so we were quite happy that the protein was active. And so we went on to try our crystallization. I mean, I don't want to bore you with all the details, but we tried various options. This is one of the nicest hits that we got from Matrix. In Index, there was another one as well. And so following classical optimization, techniques, change in pH, concentration, protein precipitant ratio, temperature and all that. We did all that. And that gave us, these very shiny plates that you can't even see well, in here. But after several rounds of optimization, these are the best crystals that we could get. And these had, in addition to the stuff, 200 millimolar glycoglycine. And then in another Hampton Index H5, there was a similar type of crystal that had 50 molar ADTA in there. At this point, we had really gotten to our weight, and these crystals are virtually transparent. And so even fetching them was very, very difficult. And so using these for structure determination was really not going to be possible. And if you were in my shoes at the time, the only thing to do is probably to just give up and then find something else doing. But again, I always remember Martin Luther in some of the wisdom that he poured out. I refuse to accept despair as the final response to the ambiguities of history. This quote was very important for me in pushing on, even when I felt that things were really very difficult. I mean, it virtually exhausted everything, and yet nothing seemed to be working right. And so one day, what I decided to do was to mix the two solutions that I had. And so I had gotten one where there was, 200 millimolar glycine, and then one that had EDTA. It didn't make a lot of sense, but I just mixed, half of the one with glycoglycine and the other one with EDTA. And thankfully, now the crystals grew a bit better. They were still plates, but these have a lot more volume compared to the initial ones that we had. And so these diffracted routinely to 2.83 angstroms. And so for the first time, we were on course to be able to at least get something out of it, which was very, very exciting. Again, because of the nature of the crystals, in terms of structure determination, there were various challenges, and molecular replacement will not work. It doesn't matter what we we did virtually everything. Heavy metal soaks wouldn't work. I mean, every classical way of phasing, we tried it. In addition to using selenium labeling, here there's a multi of the unlabeled and the labeled. You can clearly see the shift in the molecular ion, clearly showing that that's about 89% labeling. What it means that there were 14 methionines in there, and so only the first one didn't get labeled, which is understandable. And so we tried crystallizing that. Thankfully, it did crystallize, and without trying to bore you with all the details, eventually we were able to phase the structure. And crystal is in a C2 space group, and cell parameters, very interesting cell parameters. And from the anomalous signal from the selenium labeled, mutant protein, we were able to solve the structure. But that's also by merging several data sets that we had collected at different, synchrotron sources across the US. Actually, three of them, all of them with different wavelengths. Sometimes some of these things are a bit weird, but that's what worked. We refined the structure to our work, our free 20-25%. Now, before I show you what we found, these are just quick reminder. In HIV-RT, these are the subdomains that have been identified. The thumb, the fingers, the palm, connection domain, and the RNSH, and these have been color-coded accordingly. And these are the two types of architecture for all retroviruses that we knew at the time. So the right hand and then the closed configuration for the P51. So we had a P66-like conformation where it's the right hand and then the closed one for the P51. Now, surprisingly, this is what we found when we solved the structure. It resembled the P51 configuration more than the P66. And what it meant was that this was actually the inactive structural configuration. But all the subdomains that have been identified in retroviral proteins were present. For the protease, it was a monomer, which is hanging on the left here. But in addition to the protease, for fomivirus, proteofomivirus, there is additional sequence between the protease and the RT, which we call the protease extension, and that we found to be packed against the palm subdomain in the structure. The RNA domain straddles across to the C terminals, but it's moved in a way that is very close to the palm. Clearly, in the absence of a substrate, trying to minimize the surface area brings all the domains very close together that allows them to interact. The thumb is hiding somewhere there. The connection domain is somewhere there. Now, this clearly tells us that for this to be able to bind nucleic acid substrate, it has to undergo very severe conformational rearrangement. And that is going to be interesting. Again, how this is evolutionarily of advantage, we don't know yet. But clearly, what it means is that you don't want to keep the enzyme hanging there with all the active groups in the right places. That may be tempting for it to begin reverse transcription or begin activity before its time is due. So in the absence of substrate, it remains in the inactive conformation, and then when the substrate is around, it triggers the conformational change. If you take the crystal structure, you could actually do a twofold symmetry and superimpose one on top of the other and get the protease to dimerize very similar to what we know classically as the protease dimer without serious conformational or serious clashes between the two subunits. What it meant was that this structure could actually dimerize in the way it is without affecting the other units in there. There was a calcium in the solution, and that could be found chelated to the RNSH active site, so we could easily identify what the the active site of the RNSH is. If you superimpose the structure on the known structure of HIV-RT bound to nucleic acid substrate, you could clearly tell where the nucleic acid substrate is supposed to bind somewhere there. And what would mean, again, the RNCH domain will have to move all the way around the thumb, and the thumb will have to move in the opposite direction to be in place, similar to the p6 domain that I had shown you. This and all this is, very interesting. And so here you see a cartoon representation of what I was saying. You see where the RNC domain had to move all the way from to this place, and the thumb has to move from where it was originally into this position, while the connection domain rearranges in there. In the literature, again, from Bob Landon's group, they had envisaged similar movements taking place to enable the conformational rearrangement to take place. And so they thought that, well, again, in the inactive state, you see an equilibrium where the subdomains are folded into very compact, unit, and then that can open up and the rearrangement takes place, stabilized by nucleic acid to allow some of the new conformations that we see that are competent for nucleic acid binding. So again, we were very excited that, well, we have seen for the first time how an immature PRRT, would look like. But the question that we kept asking was, I mean, is this going to be similar in HIV and other, retroviruses? And here we only had the protease reverse transcriptase. So then the question is, how is the integrase going to fit in here and all that? And so we turned our attention towards HIV, try to see whether we can replicate the same study in HIV. And so looking into the literature, the Swanson group had done beautiful work back in 2005, where they show that the maturation of the gag pore, which contains both the structural and enzymatic proteins, is not a random event, and that it goes through a well choreographed process. So from the gels, you could see initially you liberate the pore domain somehow, and then that goes on to cleave the other polyproteins that are, so that intermediate polyproteins that are produced before they are subsequently cleaved into the mature enzymes. What it means is that the protease in the pore or in the gut pore has to be active, and then in the other polyprotein intermediate has to be active. But it was also found that the initial cleavage happens between P2 and nucleocapsid junction, or nucleocapsid and transferrin domain junction, thereabout. But the long story or the take-home message is that irrespective of where the initial cleavage happens, it's likely intramolecular, and the released polyprotein retains activity for the protease. And these gels show that beautifully. And so we went to work and also began working on the pore portion of the polyprotein to see whether it's going to be amenable to structural studies. Because I was working in E. coli, again, these are just reminders for people. If you want to express proteins in E. coli, these are some of the things that you want to look at. What strain, temperature, the amount of inducer, IPTG, or arabinose, whatever your inducer is, and then the type of media you're going to use. Sometimes you modulate or you crisscross these, and then you can find a sweet spot where your protein expresses well. On the protein side, you look at what type of vector, the promoter there. Sometimes the protein sequence does affect whether your protein gets expressed or not. Tags can be very important. Other binding partners, chaperones. So again, there are things that you can do at the protein side to make sure that you're successful. And so we went through all these with our initial construct. But after creating the initial construct and then trying with various media, we're not successful in being able to purify HIV1 pore, to the point where we could use it for structural studies. Basically, as you can see, there's a lot of proteolysis going on, and many of the proteolytic products could not be purified away from the full-length protein for any meaningful study. And so again, when your strategy doesn't work, you go back to the drawing board and see what is it that you can. And most of the time, you go back to the media to see, okay, what is it that we can do. So some of the things that we started doing is looking at different media compositions, to see whether any particular media was going to work, and none of it really worked in a way that allowed us to be able to purify good materials for structural studies. And so, again, I'm not going to bore you too much with all the details. I had to reformulate media that would work. I didn't know. I found out these things one after the other, or sometimes even serendipitously. But I found that having a lot of nutrient there is very helpful. So instead of 1%, I had tryptone 1.5% with by volume, for all of these in there. So regular media yeast extract 1%, sodium chloride, a bit more salt, appear to be much better. And then adding enzyme was also important somehow. Glycerol for allowing, of course, as extra energy source, but also for shaking, allowing oxygen so that we can ship at a higher speed. And then, but the game changer really was these two guys. We found out that having magnesium at least 50 millimolar and pH at 6 really reduce the proteolytic processing of our protein in E. coli. And we had the option of even add a phosphate buffer, and the protein was still fine. But we needed to have magnesium at least 50 millimolar and then pH of around 6 for the protein to work. And so we went back to our construct, which we had used earlier on using BL21D3RIR, expressing at 15 degrees Celsius. We could purify the protein by nickel, heparin, and gel filtration, and get very nice peaks from there. So again, I'm just showing you the gel filtration profile because it gives us some indication of what we were to expect. The majority of the protein appear to be a dimer based on the calibration of this column. The protein looked very nice on the gel, and from the DLS experiment, majority of the proteins were corresponded to what the gel filtration was saying. So if you look at the peaks, most of them, the molecular weight was around 234, which is about 93% of everything that's there. Our construct had a molecular mass of about 120 something, and so clearly that dimer would get you close to the 234 that we had. So at least for the first time, we had been able to purify, the protein, express and purify that, and we were ready. But we were curious as to why the pH and the magnesium worked. And so Joe and Linda in the lab at the time decided to check by Western what was going on. And so here on the left are various types of media that we had initially tried to express the protein. You can clearly see using anti-integrase antibody, all the proteolysis that you are seeing there. Now, using the media that I had just created at pH 6, you can clearly see that the proteolysis is very much reduced. They decided to remove some of the ingredients in there. It didn't help too much where you see some proteolysis. But if you increase the pH to 7 for the same media, now you begin to see more proteolysis, taking place. So clearly, the pH was very important, and that the pH 6 was a sweet spot where expressing the protein, you have reduced proteolysis that allowed you to be able to isolate these guys or this protein in a reasonable manner. So again, I've already mentioned the pH. So if you look at the magnesium side, again, was a similar thing. I'm just reproducing these as before so that you see the pH 6 reduced proteolysis. But here, if you did the same experiment, but this time around, you used 10 millimolar magnesium, now you hardly have any full-length protein. And most of them is degraded. Now, if you do the same experiment where the magnesium level is at 50 millimolar, you can clearly see that you have similar pattern to what you saw, with the pH 6. So you get a full-length protein, very little breakdown on that. And so this is when we knew that, okay, well, now we know that the magnesium and the pH are the two factors that are the most important in there. Just for the record, when I formulated the media, I didn't know what the effect of these were going to be. I just figured out that they worked. And so Linda and Joe were more curious to find out what exactly was happening. Was it really the magnesium or the pH or both that are responsible for that? And so this clearly told us that it's the magnesium and the pH actually have a similar effect. And so combining the two makes it even better. And so that is why in the media that I put here, I put the magnesium at 50 and then the pH at 6. So this is what we use for the expression that we did subsequently. And so, well, we had purified the protein, wanted to know whether it's active. And so again, we sent it to our collaborators. They came back, the protein was active. So Jeff DiStefano was one who helped us with this part of the assay. So now protein is active, purified. Because we knew that crystallization was not going to work, we tried a few things. So we went to cryo-EM. And so Eddie's long-term collaborator, Dimitri Limkis at the Salk Institute had been the person that we've been working with for some time. And so we sent the protein to his lab, initially did a whole bunch of screening, got some very good grades. And then again, we had to go through the pipeline of data collection, processing, particle picking, 2D classification, and 3D. So this was an iterative process where you pick particles, did a classification, and then eventually we created, templates from there, and I used the templates to pick more particles and rated the classifications to make sure that in the end we have high quality particles that we could use for 3D refinement and classification. And so in all, this is just the workflow that we did, and everything we did was in CryoSpark initially, and then later we had to do some other work in other software, but most of the work that we did in Dimitri Lab, we use a CryoSpark. Just before I show you the structure that we got, just a reminder, the protease functions as a dimer, the reverse transcriptase as a heterodimer, and the integrase, the basic architecture is a tetramer, but we know that forms higher order oligomers that are actually active. And so for the pole, these are the structures that had been solved at the time. And so when we looked at the architecture of the pole, in fact, all that we saw basically was the reverse transcriptase. And so we were worried that maybe the protein had broken down somehow and that we could only refine the reverse transcriptase portion to high resolution. But again, it did resemble everything we know about the HIV/RT, nothing surprising over there. But whilst we were contemplating, we went back to look at the 2D classes that we had. Now, interestingly, again, we just show a few of these particles where the red arrows are. You can clearly see that there is some density there that is unaccounted for. So clearly you can see the RT over there. And then on this side, there's this fuzzy density. And so a technique that the research lab pioneered over the years called focus group classification was used, where you try to mask the well-defined portion and then see whether the density over there pops up. And when focus group classification was done, there was a density that appeared close to the RT that matched the density for the protease. And so here we were able to recover the protease density quite nicely, affect the density of a protease, and we could actually improve it to a point where a model could be built for the fuzzy density that was there. But in terms of the details, I mean, the protease did resemble the natural or the mature protease dimer that we know. But I wanted to pay attention to this. There's a new interface that has been created. By joining the protease and the RT together, you create a new interface here. And you can see how the P51-like domain now goes to join the this P60-like protease and then the corresponding P51-like. So we were convinced that this is absolutely the right configuration, and the C-termini of the protease will be coming off of there, and the whatever additional things going to be go down through there. And so we're very excited that this clearly tells us that the protease is going to dimerize, yes, but the RT also is in the heterodimer form. Unfortunately, we couldn't recover the density for the integrase, again, because of we reckon that it's very flexible, just like the protease, and so we couldn't recover the density for the integrase, except that in the 3D classification that we did, you could see the NTD from one of the subunits. So from the P6, you could clearly see the NTD domain of the one of those integrase units protruding over there. Using a small angle X-ray scattering, we could actually also confirm that, yeah, that shows, or that is likely the integrase from the NTD of the integrase of one of those domains. So we're very excited that we could see some part of the integrase, even though we couldn't tell what oligomeric state it is in. At least clearly tells that, well, the protein is intact, and that we have gained some insight in what is happening. And so the overall question is, how is the integrase looking? So one thing that we also did was to see whether the integrase in the pore is able to bind some of the binding partners that we know. And the integrase binding domain of IBD is something that we know binds integrase. And so we co-expressed the pore with a construct of the IBD fused to a motors binding protein. And interestingly, the motors binding protein was able to pull down the pore, and on-jet filtration remains intact. So this is after 2 rounds of purification, washing the column with 1 molar salt and all that. And so the binding is as tight as one would expect. And so what it means is that the integrase is able to dimerize even whilst it is still in the pore. Of course, we have not been able to visualize the integrase in the structure, but from this experiment, we know that the integrase is capable of dimerizing, because the IBD binds integrase dimers. So we're very convinced that, well, the integrase is still there, and that nothing had happened to the sample whilst it was in transit. And so based on all these, we propose a model where in the gag pole, there is some kind of difficulty because of all the various elements that have to come together to get a protease to form a dimer. And so that initial dimerization is very sluggish. But once that dimerization happens, there is an intramolecular cleavage of the gag pole, right, between the NC and the transframe region. And then when that happens, that generates an intermediary polyproteins which contain the protease. But because RT has a very vast dimerization interface, we believe that just as other biochemical experiments have shown, the RT dimerizing helps the protease to also come together. So in the previous structures, we show that here the RT is a dimer, and if the RT is a dimer, that could actually facilitate the coming together of two protease units. So here, the RT forming a heterodimer, you have two of the proteases now close enough that they can also dimerize. So we believe that the RT dimerization could help with the protease dimerization because the RT dimer is very, very stable. And then while the protease is in there, now it's able to go ahead to go back and cleave, the various polyproteins into their mature form until the protease itself is able to cleave itself out of that, and then the process continues. So with this is the model that we come up with, where gag pole dimerization is quite infrequent, but we got all the conformational challenges have to face. But once that happens, intermediary polyproteins are produced, and therefore that process continues until the mature structural proteins and the enzymes have been liberated. But it's a clever strategy. What it means is that the stable or the active units that are needed for downstream processing don't have to be figured out again. If they through the gag pole dimerization, you are creating these active units that are going to use for downward processing, that remains a very clever way of making sure that the timing and everything doesn't get delayed because you can't find binding partners or you can't find partners and all that. So again, based on that, we're asking ourselves, well, since we couldn't visualize the integrase, we're still working with HIV2 and HIV1. Can we use FABs to be able to stabilize the flexible regions in the protein to enable us to be able to visualize where the integrase domain, for example, is? So some of the work that we've done recently is using known templates to design FABs, and then using the computational algorithms to sort of design epitopes that will bind, design CDRs that will bind the various epitopes that we have in GAG and GAG pole, because that's what we are trying to study, and then see whether we can validate these things through experimental work. For the initial stuff that we've done, well, successfully generated various antibody fragment with various different CDRs and different epitopes. Using, again, computational methods, you could see very good binding affinity between the epitopes that we designed and the FABs. For the record, for a known HIV1 FAB, using the same system, binds with about minus 8.5 kilocal per mole. And so getting minus 9.4 to 10.33 kilocal really means that there's likely strong binding. We are currently in the process of validating, expressing these antibodies, and then validating these bindings so that we can use them for structural studies. Again, after we published this work, Merck scientists published their paper showing that they have some small molecules that they believe targeted the gag pole, but these have very unique tropisms. The phenotypes are said that when they put these molecules in there, those molecules selectively kill HIV-infected T cells. And so we know that, well, targeting the gag pole may be a very interesting way of tackling HIV. After all, we were convinced that we would find new interfaces in there where we could design new molecules to target. And so some of the things that we've done, recently was to use our virtual screening on some of the new interfaces that we have realized or we've seen in the HIV-1 pole, structure. We've screened so far 10,000, and these are some of the hits that we believe that could potentially work. And so these are basically docking with some MD simulations. We are still working to validate these, but clearly you could see these, even though they are quite shallow, you can see that there are pockets in there where sometimes these molecules fit in very nicely. And we are hoping that some of these molecules would actually show activity in cell-based assays, so that we can begin to look at their structures as well to guide further design of the new generation of HIV inhibitors. So to conclude, I hope I have convinced you that adding 50 millimolar magnesium and changing the pH of the medium to 6 actually attenuate proteolytic processing of heterologous expressed proteins in bacteria. This condition allowed us to be able to successfully express HIV-1 pole and other protein that we couldn't express before in there. And that enabled us to solve the cryo-EM structure of HIV-1 pole, which showed an RT, a heterodimer, and a protease dimer, which is proximal to the N-terminus of the RT. Now, we believe that the dimerization of the RT does help with the protease activation. And so the virus is very clever at coupling RT dimerization, which is needed for downstream processing, to protease activation. And that effectively means that only one RNSH of the P51 is available for cleavage, because in the field, people have also wondered why only one RNSH domain of the P66 is cleaved, while the other one is not cleaved. So this clearly shows us that there is only one RNSH domain of the P51 that is available for cleavage, and that is why it is cleaved. Again, by virtue of the fact that the pore, the integration in the pore, whilst remaining disordered, can bind and pull down IBD domain of LEGF, suggests that the integrase can also form a dimer whilst it is still embedded in the pore. And these are very exciting. We have these new molecules that we've shown that bind to new pockets that we've identified in the structures, and we are hoping that these compounds show activity in cell-based assays. So with that, let me thank all the people who have been very instrumental in this work. Eddie Arnold, who's been always with me as a mentor and a friend. Joe Bauman, Linda Tabity, who were at the time in the lab. My very good friend, Chavi, he has been fantastic in all the things that we've been doing in terms of building a model, refining structures here and there. Without him, we could have done this work. Of course, our collaborators at ASOC, Dimitri, Lemicus, Jessica, and Dario, even though they've moved to other places, they were instrumental in this work. Of course, Jeff DiStefano, who did a lot of the assays for us. And I mentioned Steve Hughes and Paul Boyer in my talk. Grateful to them. this work was supported by these NIH grants. At the time I was in the US. Currently, my lab is funded by LifeHack through the Crick Africa network, and they are providing support for the virtual screening and computational, and then the other validation tools that we are currently working on. Thank you very much for your attention. I'll be glad to answer all your questions. Thank you.
Thank you very much, Jerry Joe. That was extremely interesting. And I have learned a lot. I didn't actually understand and fully appreciate the purpose of the dimerization and the way in which the polyprotein could form without the dimerization. But anyway, I'm going to open the floor to questions, and people can ask as they indicate. Are there any hands up? Well, people can tell me if they got their hands up. No. Okay, I'm going to ask a question then. Addy, have you got your hand up?
I think somebody's hand is up.
That's Addy Shana.
Okay, sure.
Do you want to ask a question, Addy?
Sure, yes. Thank you, Trevor and Jerry for a very interesting talk. So I'm not a structural biologist, but I just have interest to understand, to appreciate your work on the expression of these proteins. I think there's a lot to learn from the way you solved these expression issues. But I just want to ask, if you also, so you hypothesized that those were proteolytic products.
Yes.
But are they not also products of incomplete synthesis? So how were you able to tell between the two? Because they could as well be incompletely synthesized products, potentially due to translational issues. So I just want to hear your thoughts on that. Thank you.
Thank you very much. That's very interesting. In fact, you're absolutely right. Some of them may actually be translational issues. In fact, Joe had looked at some of the papers people have published on HIV integrase and found that there are cryptic start sites in the HIV integrase. So some of the lower bands could definitely be, incomplete, proteins. But because we used a C-terminal antibody, so the antibody binds at the C-terminal end of the integrase. And so if these were necessarily abortive translation, from the N-terminals, it may not get to the C-terminals before they are aborted, right? The antibody binds to, I think, the last 20 residues in the protein. So if they're actually abortive, we wouldn't see them on the Western. So that is why we thought that these are proteolytic products, because they do have the C-terminal epitope that the antibody targets. I hope I've answered the question.
That makes sense. Yeah, makes sense. Thank you.
Thank you.
Other questions? Okay. Have you made a model of the complete dimers of the polyproteins at the different stages of the development? Is that a relevant thing to do?
We have tried, but we haven't been that successful. So it's definitely something worth doing. We had a crude model, but nothing that would be useful or be important for publishing. But yes, it's very important, because we are still perplexed as to how this intramolecular cleavage happens. The whole gag-pore dimerization thing, we're still grappling with it. But even how integrase is behaving in there. So definitely, a model would help.
Thanks, Jojo. Anybody else want to ask a question right now? I don't see any hands. Oh, there's another question from Addy. Go ahead.
Thank you. Jerry, you used antibodies to infer on your protein fold. Am I right?
Yes.
How reliable is that? I mean, why did you not employ CD-SPEC or some other?
Okay, so
like
not necessarily the folding. The antibodies are not for the folding; they are for Western blot. What the additional other work that I want to use the FABs for is that there are flexible interdomains. So these proteins are made up of multiple subdomains, and in between the multiple subdomains, there are flexible regions. And so, because of these flexible regions, we couldn't visualize them in the cryo-EM. And so, what we are hoping to do is bind these FABs or these antibodies to these flexible regions to try to stabilize them for structural visualization. So, not necessarily to infer folding from them.
I see. Okay, so they are like some disordered regions.
Yes.
Okay, thank you.
Okay, with that then, Jerry Joe, thank you very much. Thank you everybody for coming. I hope you enjoyed it, and please join us in a month's time for the next talk in the series. I just want to say, Lawrence, if you stop recording, you have stopped recording. You know that we've got an offer from Cassini to give these talks a DOI and to publish them as a lecture on their website. Are you happy with that?
Yes, that's totally fine with me.
Okay, good. Cassini is part of Springer Nature.
Sure.
Okay. Okay. Thank you very much, everybody, and thanks, Jerry Joe. That was a really nice talk. Bye, Zoe. Bye, Jerry.
Trevor, are you still there?
I'm here, yes.
So I had a power failure here. And so did it stop recording?
It told me that it stopped recording, yes.
Okay. But it was also recording from the YouTube on YouTube. So that should have kept going.
Okay.
And then so you should have become host. I'm trying to figure out what happens in such situations, right?
You made me host right in the beginning, so
I made you a co-host.
You made me co-host anyway, but I could do anything.
But then when I dropped out, from what I understand from the instructions and the webinars I go to on Zoom, I think it automatically picks one of the co-hosts and makes them host.
Okay. Anyway, it was fine, except that it's told us it stopped recording.
Yeah. Okay. So we may have to stitch together the last few minutes onto the recording that we have in the cloud.
Okay.
This recording might be a little messed up. Let's see how it goes.
Okay. Let's see how it goes.
Okay, and then let me figure out why the power went out for a quick second.
Is it just at your house or is it everybody?
I don't know. I'm not at home, but it's might have been this whole hallway. I'm at the office.
Okay.
All right.
Okay then. Thanks, Lawrence.
Bye.
Thank you very much. Right then. Bye.